Good day, everyone. Doc Mika here, and today we will be discussing the process of viral replication. Viral replication is the process by which a virus particle makes a replica or a copy of itself, meaning everything that makes this virus itself must be made again. All right, so from the nucleic acid, the capsid protein, if it has an envelope, it includes the creation of another envelope, or the proteins as well. Okay, this replica must be the same with the other virus, or what we call the parent virus. It involves the multiplication of the viruses in a living host organism, right? Remember, viruses need a live host cell for it to be able to replicate. So that is from the point of infection or the entry of the virus until the release of new viral particles from that infected host cell. So this process was first studied in bacteriophages, and that led to the description of the one-step growth curve or the one-step multiplication curve, right? Remember, viruses are not replicating through the method of other unicellular microorganisms like bacteria, protozoa, or fungi, which is like mitosis and binary fission. Viruses, okay, just to for you to visualize it, the experiment that they did. All right, so look at the graph on the lower left of the slide. The point one, okay, the first point was inoculation. Okay, they have a cell culture and they inoculated it with virus. Okay, with bacteriophages, and then they were able to see the number of the infectious variants in different time periods of the experiment. At inoculation, they see that there is a decrease in the virus that is detected extracellularly or intracellularly. And we enter a period of what they call eclipse, meaning this is when the virus, the entire particle, disappears. It cannot be demonstrated as it is. This is when the virus enters the cell, gets disassembled, and the process of replication happens. And after that, after the eclipse period, there is a burst. Suddenly, the host cells release many viral particles. That point, okay, from inoculation to the release of viral particles, that is what we call the latent period. Okay, the virion, the virus particle, can now be demonstrated again in record numbers, intracellularly or extracellularly. So let's get into what happens in that eclipse period. What happens during the stages of replication? As I have repeatedly said, viruses need host cells to replicate. They commandeer or hijack or take advantage of the host cell machinery to produce more virus particles. Remember, the virus does not have its own metabolic machinery, so it uses everything inside a cell to help it replicate itself, right? So we have a variety of stages. It starts with a virus attachment on the cell membrane of the host cell. It enters. The virion is disassembled, releasing the genome to the site of replication. And this replication, we have discussed it a bit in the previous lecture when we discussed the Baltimore classification of viruses. And this genome will be replicated for it to be, one, translated into the viral proteins, and for it to be replicated to produce more copies of the genome. And these viral components are assembled and later released in a variety of ways. Okay, so let's take it stage by stage. Attachment. This is the first stage of viral replication, the viral attachment proteins, meaning yung glycoproteins na nasa either envelope ng virus or the capsid protein itself in naked viruses, binds to the cell receptors on the host cell surface. These cell receptors can be made of a variety of macromolecules, carbohydrate, protein, or lipid components. The most common is glycoprotein. On the other hand, the ligands on, I said this already. Ligands can be the envelope glycoproteins for envelope viruses, or for those viruses which lack an envelope, it would be the capsid proteins. The attachment starts as a weak ionic bond, with low affinity. Hindi siya ganun ka-strong. However, other receptors on the virus particle and on the cell bind, which strengthens this initial bond. One important thing to remember is that a virus will only bind to a cell with the appropriate receptor. For example, an HIV-1 will look for the CD4 receptor, which is commonly expressed by immune cells like the T helper lymphocytes, macrophages, and monocytes. The poliovirus will look for the PVR, or the poliovirus receptor. The Coxsackie B virus and adenovirus share a receptor, which is the CAR, or Coxsackie adenovirus receptor. All right, so this is unique for every virus. All right. Now, this specificity of the virus towards its host cell, yung pagiging maarte ng virus pagdating sa target cell na i-infect niya, this is what we call cell tropism. All right? And this is not just for cells. From cells to organ tropism, tissue tropism, and species tropism. All right? Kaya may mga virus na it only infects dogs, or it only infects monkeys but not humans. All right? This stems from the molecular morphology of the virus and the protein that it needs to bind to. Okay? So for example, the HIV virus has a protein on its surface, which is the GP120. It will look for the CD4 protein expressed on the surface of immune cells. For the influenza virus, the hemagglutinin glycoprotein would look for the sialic acid in pneumocytes. The transmissible gastroenteritis virus protein would target the aminopeptidase N in enterocytes. All right? So if hindi niya mahahanap yon, for example, in this image here, which is this is a canine distemper virus. All right? It will look for the canine blood mononuclear cell. And the protein there is SLAM. Okay? In the table, I think it's there. Signaling lymphocyte activation molecule. And they would fit. Okay? And that would initiate the process of replication. The virus could go into the cell and start replicating. However, if it goes in a feline body, it will just bounce off and it will be shed. All right? So that is basically the basis for cell tropism. And the thing is, viruses can get a little bit smart, Actually, they're very smart. Because there are diseases right now, viruses, which have a large cell and organ tropism. Meaning, it can infect a lot of kinds of cells. From fibroblasts, keratinocyte, melanocytes, osteoblast, myotubes, macrophages. So if you expect as well that the clinical symptoms that you will detect, that you will see, is of broad range. General, hindi lang siya diarrhea, but you will see neurological signs, you would see hormonal signs. So these are the viruses that are very tricky to detect and very tricky to treat as well. This is the chikungunya virus, one example. A mosquito-borne disease, right? But once the virus attaches, okay, what happens? How does it penetrate the cell membrane? Remember, the cell membrane of the host cell, the epithelial cell, is around 7 nanometers. It may be thin, but it's very good in protecting the cell from anything bad getting into it. So what does the virus do to defeat this protective function of the cell membrane? Okay? So, depende sa kind ng virus. Okay? There are two general ways or mechanisms for viruses to enter the cell. Okay? One is direct fusion. The second one is endocytosis. Okay? For enveloped viruses, commonly they use the direct fusion method, wherein they shed the viral envelope and the cell membrane fuse, and it will release the nucleocapsid inside. For receptor-mediated endocytosis, same thing as when molecules that the cell needs enters through endocytosis, like phagocytosis, spinocytosis. The difference here is that the glycoproteins in the viral envelope or the viral capsid would attach to a receptor, and that causes the cell membrane to invaginate and coat this virion and later be released. Okay, we'll discuss this in detail as we go along. All right, let's talk about direct fusion. Direct fusion is when the viral envelope and the cell membrane fuse, forming a fusion pore or a hole through both membranes, which will be used by the nucleocapsid to enter the cell cytoplasm. But how does this happen? We know that the plasma membrane is an effective physical barrier. The virus particle is stable enough as well. So what could make both membranes of these two structures break and join? All right. The viral membrane, in this illustration, okay, the upper part labeled CM is the cellular membrane. Okay, the VM would be the viral membrane. It could be the capsid or the envelope. It depends on what kind of virus. Okay, and this viral membrane, okay, contains what we call a fusion peptide. The fusion peptide, which is yellow in this case, the yellow small rectangular pair there, is in a metastable state or the pre-fusion state. Okay? Dito sa state na to, it's stable. Okay? The proteins always want to be in a stable position. Okay? So, what makes it unfold? Okay? This peptide, which is surrounded by other fusion proteins, can be activated by receptor binding. When this fusion peptide detects that the receptor of the virus and the receptor of the cell binds and forms, a bond, that ionic bond, this fusion peptide unfolds and anchors to the membrane opposite it, the cell membrane. Okay? It undergoes a conformational change and anchors it. All right? Now remember, I don't want to go to the molecular part of it, but remember these areas, the middle part of the cell membrane and the viral membrane, are quite unstable. Okay? They have a hydrophilic end, a hydrophobic end. Okay? What happens when the fusion peptide anchors is that a hemifusion stalk hemifusion stalk is created. Okay? The less stable conformation ng fusion peptide, kapag nag-unfold siya, it would want to fold again. So what it will do is it will force the membranes, okay, the outer part of the cell membrane, the outer part of the viral membrane, to mix and fuse to form the hemifusion stalk. Okay? This fusion peptide is so strong that it wants to fold. It will bring the cell membranes to fold with it. Okay? And once it is folded completely, this will form the fusion pore wherein a virus could enter through. The nucleocapsid specifically. And this is true for example, paromyxoviruses and retroviruses, okay? And the thing is, this, what do you call this? This fusion pore, okay? It's not as specific as the receptor, but it can be activated by the simple receptor binding, okay? This is a graphic for that, but the direct fusion happens so fast. So you can see that is a envelope virus. The cell membrane is in the lower part. It binds. Okay. All right. Causes the membranes to fuse. See, it's fast. And to release the nucleocapsid inside. And later on, the nucleocapsid will be disassembled, releasing the genome. All right. So that is just one way for viruses could enter. What's the other one? The receptor-mediated endocytosis. Okay, in a receptor-mediated endocytosis, okay, the virion particle binds the receptor, starts as that. However, this binding would lead to the invagination, okay, of there are already invaginations sa cell membrane, okay? It's for the normal physiological process of endocytosis, and the virus would be contained in a vesicle lined with a coat protein. The coat protein varies depende sa virus. It could be clathrin, it could be caveolin, it could be pinocytosis. All right. So let's watch this video for that.
Binds to a specific receptor on the cell's plasma membrane, forming a ligand receptor complex. Each receptor is associated with a protein known as a clathrin on the membrane's cytoplasmic side. The regions of the plasma membrane in which these receptors and their associated clathrin molecules are expressed are known as clathrin-coated pits. The clathrin-coated pit sinks into the cell, forming a vesicle that contains the ligand receptor complexes. Once in the cytoplasm, the clathrin molecules coating the outer edge of the vesicle leave and associate with new receptors on the plasma membrane. The uncoated vesicle fuses with an endosome, and the ligands and receptors separate, collecting at opposite ends of the endosome. Sections of the endosome containing unbound receptors pinch off, forming transport vesicles that return the receptors to the plasma membrane. Enzyme, smaller molecule plasm of the cell for use in a number of cells.
This is just
binds to a specific receptor on the cell's plasma membrane, forming a ligand receptor complex. Each receptor is associated with a protein known as a clathrin on the membrane's cytoplasmic side. The regions of the plasma membrane in which these receptors and their associated clathrin molecules are expressed are known as clathrin-coated pits. The clathrin-coated pit sinks into the cell, forming a vesicle that contains the ligand receptor complexes. Once in the cytoplasm, the clathrin molecules coating the outer edge of the vesicle leave and associate with new receptors on the plasma membrane. The uncoated vesicle fuses with an endosome, and the ligands and receptors separate, collecting at opposite ends of the endosome. Sections of the endosome containing unbound receptors pinch off, forming transport vesicles that return the receptors to the plasma membrane. The remaining
example of that receptor, the creation of the complex, invagination. This will later further invaginate. All right. Oh, damn, that's slow. That is slow. Okay, let's watch it anyway. All right. This will break off from the original cellular membrane. This, the contents of this, the coat protein will be shed. It will return to the cell membrane. And this process, okay, varies from different viruses. it is said that the vesicle would fuse with an endosome and then later would fuse with a lysosome, which would degrade the membrane inside and release the contents into the cytoplasm. Right? We'll discuss this in detail a bit. All right. So fuses with endosomes and sorry, it's nighttime. My head is not working. All right. And this one, as I've said earlier, could be through pinocytosis, phagocytosis, and receptor-mediated endocytosis, which utilizes different proteins. This is where, you know, the the magic of virology happens, because they know the proteins depending on the virus, which is cool. But we are going to discuss this more per virus. Okay? Kasi naman i-enumerate ko lahat yan ngayon, then makakalimutan nyo na lang later, diba? Okay, so what's the next step? The virus attached, it was able to enter the cell. What happens next? The nucleocapsid, okay? Remember, it still has the nucleocapsid, must reach the site of replication. And the site of replication can be different. It's among viruses. It could be in the nucleus or it could be in the cytoplasm. And the thing is, the nucleocapsid cannot just release the viral genome it's protecting if wala pa siya dun sa site of replication. Okay? It cannot just say, okay, bahala ka nang pumunta kung saan ka magpaparami. Okay? They're very smart like that. Okay? It utilizes what we call the intracellular trafficking system. Okay? Remember, cells, it doesn't just have organelles, diba? Ano pa ang laman ng cells? It has microfilaments. It has microtubules for the transport of different proteins that it needs inward and outward. And the virus utilizes that network. Okay? And make sure that it utilizes it in a way that is beneficial for it. So I need you to follow me through this, okay? This animation is hell. Alright? So, alright. Two places lang naman yan. It's either the nucleus or the cytoplasm. Okay? So what are the options of the cell? Now, this differs among viruses. I just want you to see how it happens and to enumerate to you. Para pag sinabi ko siya later, alam niyo na agad. Hindi ko na kailangan sabihin to lahat. Okay? So, this is a cell. You have the nucleus there, pink circular one. It has pores. The NPC is what we call the nuclear pore complex. Okay? Yan yung normal na mga, kumbaga, pinto ng nucleus for things that needs to come in and for things that need to come out. Okay? But that is password protected. Okay? You need to have the password to go in. Alright? Especially if you're quite big, sometimes it lets in some particles which are small enough to get through the door. Okay? Like imagine a door, may lock siya, pero you're small enough to creep under the door. Okay? Works that way. All right, let's go through the options. You have a virus outside the cell. It enters, okay, and it sheds the envelope. For example, this is a receptor-mediated endocytosis. It is now covered by a vesicle, okay, and it makes sure, okay, may target siya. Pupunta ako ng nucleus, pupunta ako ng cytoplasm. Imagine the intracellular trafficking system as a railroad, okay? And this virus is riding a train. It has to make sure that the train it is boarding goes to where it needs to be, okay? I think that's the best way to visualize this, okay? So, the virus particle is now released from the vesicle. Remember, diba? The endosomes will release the nucleocapsid into the cytoplasm. Now, there's a lot of options where the nucleocapsid will go, okay, depending on the virus. First, it could go near the nucleus because that's its target area, and it could do three things. Okay? Three things. The nucleocapsid can shed the capsid protein, release the genome near the nucleus. Okay? Follow the animation. That, again, you need a password for that. Sometimes you just need a password. There are proteins around the cytoplasm, and it differs for every virus. You have dynein, kinesin, indocin, importin. It's different, so I'm not gonna identify it here. Okay, that one binds with the viral genome. Okay, the viral genome just knows what protein it needs to pass through the nuclear pore complex and go in and multiply. Okay, what's the other option? Right, it multiplies there. The other option is that the nucleocapsid itself, right, could go in the nuclear pore complex. This is yung mga maliliit na mga viruses. It could just simply go in. It will shed the nucleocapsid inside, release the genome inside the nucleus. Another option is that the nucleocapsid will dock. Kumbaga, magpapark siya sa harapan ng pinto. Okay? It will recruit another protein, an import protein with a password, shed the nucleocapsid, and enter. Alright? Tatlong options pa lang, ha? Tatlong options. There's another one which I did not put here, wherein there are viruses like the parvovirus, wherein they cause disturbance along the nuclear membrane. Okay? Honestly, the molecular part of how it causes, that havoc inside the nuclear membrane for it to become unstable, right? And the parvovirus makes that big disruption around the nuclear membrane, causing the nuclear membrane to develop gaps. Okay? Nagkakaroon siya ng butas. And the parvovirus takes advantage of that. They will go into the cell and start replicating. Another option is that some viruses don't need to go into the cell. Okay? They just need to go into the cytoplasm. It sheds its it sheds its capsid and goes straight into the ribosomes for translation. Okay? This is true for those RNA viruses. All right? And that's where they multiply. So these are simple the simple mechanisms as to how they hijack the normal cellular proteins and microtubules, microfilaments for their own gain. Okay, that's how smart these viruses are. Okay, another example of that, as you can see, the polyomavirus could go in, the adenovirus parks outside the nuclear pore complex and sheds the capsid as the genome inside. The herpes virus, pretty much the same thing. It's just the matter of gaano ka sira yung nucleocapsid nila. Okay? Again, we'll discuss this in more detail as we go to every family, which is hell. But it's good. Okay, what happens now after they shed the viral genome? Okay? The viral genome now starts replicating, and I've discussed this in the previous lecture. Okay? This is the way to remember them, the Baltimore classification. But I'll give you an example anyway, a summarized example anyway. What happens in the replication part? Remember, ano ba yung kailangan nating i-replicate? The virus particle. Ano ang nagme-make up sa virus particle? At least the viral genome and the capsid protein. So we have to create more copies of the genome and more copies of the protein. So let's discuss transcription first. How we make more genomes or nucleic acid. For DNA viruses, they replicate in the nucleus except for pox, asfar, and irido. Okay? Everyone replicates in the nucleus except for these three. Now, for the nuclear double-stranded DNA, they use their own DNA polymerase for these viruses. For the exception to the rule, which replicate in the cytoplasm, they use the viral DNA polymerase. They carry their own enzyme for transcription. And for the two viruses na laging exemption to the rule, the ssDNA, the single-stranded DNA, parvo and circo, they use the cell's DNA polymerase. Okay? It's pretty much the case. Most often, when they replicate in the nucleus, they use the cell's own replicating enzyme. Right? For RNA viruses, they replicate in the cytoplasm except for the retroviruses and the orthomyxoviruses. Okay? So what's the breakdown for this group? Okay, it's kind of wordy for now, but this will help. For positive sense ssRNA, they directly bind to ribosomes, as I discussed earlier, without need for transcription because they're already the correct format for the mRNA. They use the viral RNA polymerase. Okay, these viruses. For negative stranded RNA, they use same viral RNA polymerase. Also, the other name for that is transcriptase. Right? However, in retroviruses, which carry a reverse transcriptase, that reverse transcriptase is theirs as well. Okay? So, after producing more genome, we now need to produce proteins to wrap the genome in. Okay? That is called translation. So, this happens in the ribosomes. You need the mRNA for that. But ano ba yung mga unang protein na ginagawa ng virus? Is it automatically the capsid? No. It first creates or produces, synthesizes, the functional proteins. What are these proteins for? This is for the initial part of replication. Number one, you downregulate the normal cellular processes. The normal proteins, okay? The normal function of the nucleic acid, the normal function of the normal output na kailangang gawin ng ribosomes, you downregulate it. You regulate the gene expression. You produce the enzymes needed for replication. You produce oncoproteins, which starts the process of neoplasia and cellular transformation. Virokines, which acts similar like cytokines, okay? But it modulates the progress of infection, meaning it affects more the infected cells rather than, the non-infected cells other than the infected cells already. And other proteins which influence the viral virulence, host range, and tissue tropism. After these functional proteins are produced, the structural proteins naman. Okay, these are the proteins which make up the capsid, the envelope glycoproteins, such as what you can see here in the SARS-CoV-2 virion particle. All right, now, na-replicate na. Okay, dumami na sila. What happens next? You need to assemble them. Assemble them in the correct way of packaging as the parent virus. Because if you do not package them the right way, they might not be infectious at the end. Okay? You have to make sure that you package them to form infectious particles. But what triggers this? What tells the cell or tells the proteins inside the cell, hey, you have to package them now. Okay? The trigger would be the accumulation of high concentrations of the viral genomes and the proteins intracellularly. Masyado nang marami tao dito sa loob, kailangan na natin silang igrupo para palabasin na sila. Okay? And the thing is, the assembly of the capsid and the genome, okay? Because you package the genome, some are segmented, some are not. So kailangan mo silang pagsama-samahin. This could happen at the same time na magkasabay or mauuna yung isa and then susunod yung isang process. Okay? What do I mean by simultaneously? Okay? They could happen just like that. Okay? This is the case for an adenovirus. Okay? Now, you could have another set of proteins and genome protein na mauunang magawa yung capsid. Okay? That's the capsid. Forgive me. Alright? And the genome is later inserted inside the capsid as they go through a capsid pore. Okay? This is what you call a procapsid, yung wala pa yung genome. Once the genome goes in, that is now your complete virion particle, which is the case, for example, the picorna virus. Okay? So, gawa, and then ipapackage na. It's the factory. It's like a factory. What happens next? We need to release the virion. Okay? The cell just says, I can't take it anymore. Masyado na silang marami dito. You need to go. Okay? But how? How does the cell have doors to just, open? They actually don't. But the thing is, the virions inside make ways for the cells to just burst or lyse to release the virions. Okay? That is one mechanism. Okay? And this is utilized by the naked viruses. Okay? Those viruses without an envelope. But how does this happen? How does the virus tell the cell burst? Okay? Just burst. Okay? Number one would be the viroporins. Okay? These are viral membrane proteins which makes pores in the host cell membrane. And it doesn't make, hindi niya ginagawang butas lang agad, but it makes it more permeable. It removes a certain protein, it inserts itself, and then it makes that small butas even bigger for the virions to go outside. They're not alone though. There are other lytic proteins, these lytic proteins, which are very, I didn't bother to enumerate the proteins because they're like CXR4GI, those terms, so I didn't want to trouble you with that. So they're lytic proteins. They facilitate cell exit by disrupting the permeability of the membrane, or they could actually induce the cell to undergo apoptosis. They could also suppress apoptosis if they think they need more virus particles to be made. Okay? Kumbaga, gusto nang mag self-destruct ng cell. Sasabihin niya, nope, make more. Okay? Magpagod ka muna, then saka mag-lyse. Okay? Very smart. And of course, another, the most basic way is that the cell itself cannot live anymore. Okay? The virus has hijacked its normal cellular functions. It cannot function anymore with its normal proteins. Okay? So it will simply lyse. Alright? That is one mechanism. The other mechanism, which is utilized by enveloped viruses, okay, is what we call budding. Alright? This process is when the virion, okay, gets its envelope by budding through a cell membrane. And it could be the external cell membrane, it could be an internal membrane, which could be from the endoplasmic reticulum, Golgi apparatus, or even the nucleus. Okay? And and before they actually decide to, just push through, the glycoproteins, which are normally, the glycoproteins of the virus has slowly deposited itself along that cell membrane. Pinapalitan niya yung normal glycoproteins na nasa surface ng cell. Okay? So it will replace those and it will recruit these nucleocapsids to get near the cell membrane for it to push through. Now, this is not like a classic lahat sila nag-undergo ng ganyan. Sometimes some nucleocapsid goes into the cell membrane with a nucleocapsid intact already, kumbaga gawa na siya, naka-package na. But remember, sometimes this process can happen ng sabay o kaya ng unti-unti. Like for example, in a retrovirus, okay? The genome could go near the cell membrane, the capsid protein could go near there and complete the packaging as the virion is released. Okay? Now this varies across different viruses again, but I'm just showing you the options for them. There's no one thing for viruses. They follow a certain, normal mechanism. But iba-iba pa rin sila when you get to the nitty-gritty of it, when you go specific, when you go to the specific viruses. Okay? What's next? All right. The preformed nucleocapsids localize to the appropriate cellular membrane, diba? As I've said earlier. However, what makes them be released? Okay, what makes the budding complete? Kumbaga. Now, it could be the interactions between the capsid protein and the envelope glycoprotein. It could be interactions between the protein and the lipid components of the cell. But most viruses recruit the endosomal sorting complex required for transport pathway, or the escort. Okay? The escort is there already for normal cellular functions of pinocytosis, phagocytosis, of releasing certain proteins outside and such. So this is a normal component of a cell membrane. What does it do? Okay, what does it do? Along with the capsid protein and the envelope glycoproteins, again, this is a teamwork, kumbaga. It just needs a little help from the cell itself. Okay, a set of proteins from the escort pathway, and they're just named escort 0, 1, 2, 3. Okay, to make it easy. This is just 4 proteins, escort 0, 1, 2, 3. They would, kumbaga, they would populate this area. Okay, they would populate this area, cause the cell membrane to stretch outward and evaginate. What's the opposite term for invaginate? to project outwards, okay, slowly. This is started by escort 1. And escort 3, okay, the yellow, looks like a tapeworm guy there, okay, causes the neck of this one to slowly constrict, okay? It forms a vesicle. That escort 3 will make a conformational change, okay, will undergo a conformational change and constrict the vesicle. And another protein, which is an enzyme, disassembles the escort machinery as soon as that neck is closed, and that causes scission, okay, and releases the vesicle. Okay, that's one way. I could simply say it just buds out, but then I always ask myself how. So that's why I research it too, because I was told why they have to teach them how it goes out. It goes out, simple as that. I myself am not satisfied with that, because I'm interested. How does it happen, diba? So another way for viruses to go out is exocytosis. Now, this is used by the viruses which get their envelopes from internal membranes. Okay? After they get their internal membranes from either the endoplasmic reticulum, the Golgi apparatus, or the nucleus, they enter the secretory pathway of the cell. They get enclosed by in an exocytotic vesicle. Pretty much how they get in, during the receptor-mediated endocytosis entry. Basically, it's just a reverse of this. And they get transported to the cell surface, where they are released when the vesicle membrane holding them will fuse with the cell membrane outside, thus releasing them into the world. All right. So that is the process of replication. Now they're out and about to infect other cells. Now, where do they actually go out of? Remember, cells have different surfaces, right? You have an apical surface which faces the lumen of an organ, especially the intestine, for example, and the basal surface which faces the blood vessels. So different viruses have their predilections as to where they would bud from. Some viruses would bud from the apical surface of the cell, which is commonly done by viruses which hit the respiratory, genital, and intestinal systems, right? As you can see there, influenza virus, paramyxovirus, and such, right? Some would go out of the basal surface. And these are the viruses which actually lead to systemic spread agad, since they can spread through the bloodstream or the lymphatics when they bud out of the basal surface. And some are complex, actually. If you think maybe they could bud from the lateral part, they wouldn't really bud from the lateral part. However, some viruses, they will bud from the apical and the basal, but they will stay there. Right? They will stay there. They would not, go out with the intestinal contents. They would stay there, and with the expression of multiple glycoproteins and such, they would cause cell-cell fusion, or what we call as syncytia. All right. One last thing that I want to tell you is about maturation. Some viruses reach maturation instantly after it is being assembled. Some reach maturation after it is shed out of the cell. Okay, it differs. But why do they need to mature? Okay, virion particles need to mature for it to be infectious. They need to be activated. Something needs to happen in their conformation for it to be infectious. All right. Example: the influenza A hemagglutinin protein, it needs to be cleaved into two fragments, HA1 and HA2, for the proteins to be capable of membrane fusion. Okay? Because if they remain in their intact, non-fragmented form, okay, they would not be able to start membrane fusion in the next host cell. And the process of assembly, release, and maturation could overlap depending on the virus. Okay, so this is just a rough sequence of events as to how things happen, so that later on, when we discuss the specifics of every virus, mas mabilis na, because I don't need to discuss the normal mechanisms anymore. I just need to tell you the the individual idiosyncrasies of viruses. All right, that is all for now from me. That is viral replication. Thank you for watching. I'm sorry if it's a little bit, I don't know. I know when I'm very well versed with a topic, but I'm learning with you guys, so thank you for listening. If you have any questions, if you have any, if you research something that's new, let me know about it. All right. Have a good week. I'll see you next week. Bye. All right. What causes the endosome to release the nucleocapsid inside? Okay, endosomes have a mildly low pH, an acidic pH, and that is the trigger for this endosome to release the nucleocapsid by fusing with the membrane of the vesicle. All right. We are going to discuss this in specific details as we discuss each virus, okay? Because it is different in every virus, in some viruses. But at least you know how it happens generally. With the specific proteins, with the specific time, with the specific names of things, we will discuss that as we go along the semester.